Okay. All right. So, yeah, as I was just saying to Bob, I've got about, let's say, 30 minutes worth of slides, and then I'll leave some time at the end for questions and just a broad discussion. And I thought I'd take you through, what in my mind is 100 years of development of TB vaccines. Really to highlight that we do seem to be at a place where certainly within the next decade we will have a licensed new and effective vaccine. But I want to take you through the history, and also the need for a TB vaccine, because it's important to understand that the type of TB vaccine that we end up with may or may not be the ideal vaccine, and maybe we'll need more than one product, or maybe then a newer product with improved characteristics. But we can talk about all of that at the end. So to kick off then, assuming I can get my slides to there we go. Okay. So this story of why we need a TB vaccine, it begins at least 10,000 years ago. Back to the earliest evidence of tuberculosis in humans. And we don't have molecular data, but we have paleopathological data from, this is a cave complex in France from about 10,000 years back, 8,000 years BC, where we have the remains from of bones, sacroiliac and femoral bones, with evidence of a chronic osteoarthritis and a femoral cold abscess, consistent with classical features of a chronic TB osteoarthritis, suggesting that this relationship between TB and humans has been ongoing for millennia. It's only quite some time later, though, about 1,000 years BC, that we have molecular evidence of that relationship. So here we have data from, again, this is a paleopathological or paleoarchaeological study of mummies from ancient Egypt, which were studied to see if the DNA of M. tuberculosis could be detected from both normal and abnormal spinal tissue. And you'll see that indeed it was. There were bones that had that classical tuberculous spondylitis, in which 2/3 of them returned MTB DNA. But in just over 10% of normal bones, also M. tuberculosis DNA could be found. Now, we don't know whether these adult Egyptians from 1,000 years BC died of tuberculosis, but we know that they died with tuberculosis. And assuming that at some stage they had infectious pulmonary as well as bony disease, then there's a high likelihood that while living, they transmitted their tuberculosis to others, and then continued the cycle of transmission, both to young and old. And this is the characteristic of the relationship between humans and TB, right? Is that TB doesn't kill immediately. It remains latent for a long time. Even when it causes disease, the disease tends to be chronic and relapsing, but it allows time for transmission. And it's against that setting that we're seeking a vaccine. Of course, TB does kill people, and yeah, we've got evidence from the mid 1600s that we've got scientific reports or academic reports here from the bills of mortality of the City of London from 1632, where it's just over 9,500 deaths, 1,797 were to be due to consumption, which is what they called tuberculosis in those days. So 19% of all deaths. In modern times, so during the COVID epidemic, SARS-CoV-2 took over tuberculosis' spot as the primary killer of humans, the single most lethal pathogen. M tuberculosis has almost certainly regained that top spot with 1.25 million TB deaths in 2023. So this is where we are today. We've seen over the last decade a very slow fall in the number of tuberculosis cases, primarily driven in people living with HIV by rollout of antiretroviral therapy. And then we see this little blip and actually an increase in TB cases straight after the COVID-19 epidemic. And we see that to a certain extent for TB deaths as well, where we've had this slow and steady fall, again, primarily driven by people with HIV and driven by improvements in HIV therapy, but also to a certain extent in HIV negative people with this little bump just after the COVID-19 epidemic. But the short story is there's still one person dying of TB every 25 seconds on this planet. So how are we done then in trying to bring that to an end? Well, we set ourselves goals, targets, milestones, and the End TB strategy that was put in place said that by 2025 we would have a 50% reduction in TB incidence rate as compared with 2015, and we're only at 8.3%. And we said that we'd have a 75% reduction in TB deaths by 2025 as compared with 2015. We're a little bit better. We have a 23% reduction, but again, much of that driven by improvements in HIV care. But although there have been considerable advances in TB diagnostics and therapeutics, those advances essentially benefit those who already have TB, right? And have some but a limited impact on TB control. And for that, we need a vaccine. So you might say, but we already have a vaccine. And of course, you'd be absolutely right. We do have a vaccine. We have a vaccine called BCG, and BCG was developed in the early 1900s by these two gentlemen, Albert Calmette, who was a physician, and Camille Guérin, who was a veterinarian, and they worked in the Pasteur Institute in Lille, France, where they cultured a virulent Mbovis strain from a cow with tuberculosis mastitis, and they cultured it again and again and again in various media until it appeared to lose virulence in animal models. And they tested this resultant strain in guinea pigs, in rabbits, in horses, and monkeys, and it did not cause disease. And eventually in 1921, so just over 100 years ago, they declared that this was a strain that would not regain virulence as a human pathogen and was potentially safe for use as a vaccine in humans. But as you can imagine, it's a big jump from declaring that you think you have a safe pathogen that will not cause disease and actually testing it as a vaccine. But remember that 100 years ago, there wasn't actually much else that could be done to protect humans from TB. Remember also the astronomical infant mortality rate due to tuberculosis in exposed children. So it was first tested in a single child, in an infant who was born in a household where I think the mother had died of tuberculosis, and then the newborn infant would have had to live with a grandmother with TB, which in those days was essentially a death sentence. And there's this wonderful description of the first use of BCG in this newborn child who received 3 doses of BCG orally, had no immediate adverse effects, and was remained entirely well without developing TB. But the early days of testing of BCG were very much this sort of random accumulation of individuals or small case series. And it was only about 10 years after this that a case series sufficiently large enough to be instructive was put together, when they'd accrued a group of just over 8,000 TB exposed children under the age of a year, and then compared those who'd received BCG compared to those who had not. And they noted that the all-cause mortality rate in these TB-exposed children was only just under 5% in those who'd been BCG vaccinated, as compared to almost 25% in those who were unvaccinated. And the interesting thing here, of course, is this is not TB mortality. This is all-cause mortality. And if there's time at the end, maybe we can talk about the non-specific effects of BCG. But certainly this was very encouraging for the proponents of BCG, despite the fact that there were many who were skeptical, and particularly about the risk of reversion to virulence. But it's only decades later that the kind of clinical trials that we would all recognize began to be done. So in the late 1940s, early 1950s, and here is one such by Sol Rosenthal. And this was a trial done in Chicago. It was a controlled trial, but without a placebo. And you're looking at the BCG vaccinated children, are the little circles at the bottom, or below the age band, and then the controls who did not receive BCG above that line. There are a couple of things to take away from this. The first is that in young children, the majority of TB cases occur under the age of 5. Well, let's say under the age of 6 here, which still maps to what we see today. The second thing is that there were far fewer TB cases amongst the children who received BCG as compared to those who were in the control group. And in fact, the third thing then is that there were no cases observed in children younger than 2, which is the age associated with the highest risk of severe morbidity and death due to TB. So these early trials of BCG in infants showed convincing protection against TB. But of course, we always want more data, and eventually we're able to do systematic reviews. And here's one such of TB cases occurring in clinical trials, which overall showed 74% efficacy against TB, 64% efficacy against TB deaths, 64% efficacy against TB meningitis, and 78% against disseminated TB. So convincing evidence from multiple case control studies and randomized controlled trials that BCG protects against both all forms of TB and against the most severe forms of TB in children. So that's the reason, largely, why BCG has been our licensed vaccine for more than 100 years. But of course, times have changed. And BCG was first used when this Model T Ford was brand new. And none of us are driving cars that look like that anymore. Other technologies have moved on, as have vaccine technologies. So let's look at what the future looks like. But first, I want to make the point that as we stand now, BCG is used in all the countries in pale blue, where universal BCG vaccination of infants is performed. And that map looks curiously very similar to this map of the globe that shows you where the tuberculosis incidence is highest. So through all the countries of Africa and Asia and into South America, where TB is most common, those are all the countries that for decades have practiced universal BCG vaccination of infants. So it is apparent that despite BCG protecting children against TB, it is not having much impact on the TB epidemic. So why is that? Well, the answer is not that BCG is not efficacious. It is. We saw in that randomized controlled or that systematic review of randomized controlled trials. It provides more than 70% protection to all forms of TB in children. The problem with BCG, and here we see in the majority of clinical trials in infants, with one or two notable exceptions, in the majority of trials, the protection offered by BCG against acquisition of tuberculosis or progression to tuberculosis disease fades by about 10 years of age. So what we're seeing is that young children are protected, particularly against the most severe forms of TB disease and death, but that those who survive through into adolescence and adulthood are or remain susceptible to TB. So this is the problem. BCG works, but it is the durability of protection is probably no longer than 10 years. So here's our situation, whereby infants who are protected against, possibly also against infection, but certainly against disease, have little or no protection by the time they get to it at late adolescence and adulthood. And that's important, obviously, because it's that adolescent adult phenotype of cavitary TB that's responsible for ongoing transmission, both to adults as well as to other children. So that's our major challenge for TB vaccine development. So what would our ideal TB vaccine look like? Well, the WHO has given us a set of preferred product characteristics. And what I'm showing you is the preferred product characteristics for strategic priority number 2. And it's number 2 because, as we've just seen, the priority number 1 is a new vaccine for adolescents and adults, and we'll talk about that in a moment. But it has described what a new vaccine for use in infants should look like. Like BCG, it should prevent all forms of TB, but including the most severe and disseminated and meningitic TB. WHO proposes that efficacy should be equal to or greater than 80%, but of course, no one's ever going to do a clinical trial with a placebo, so we're never going to observe that. or superior efficacy as compared to BCG, which is a high bar, because as we've seen, BCG is at least 70% protective. So we're proposing here that we would want something that statistically is superior to 70% efficacy. In terms of safety, it should be as safe as BCG, but with demonstrated safety in infants with HIV, and ideally without the quite severe reactogenicity that we're all aware of. Strategic priority on number one then is the new vaccine for use in adolescents and adults, and that's priority number one because this is the form of disease that drives transmission. So that's the indication: prevention of active pulmonary TB in adolescents and adults, ideally with 50% or greater efficacy. And we would ideally like it to prevent TB in people who at the time of vaccination are both TB sensitized or not sensitized. So we'll talk about that in a minute. Who live in all parts of the world, and it should be safe in everyone, but also in particular risk groups such as people living with HIV. We'd ideally like that vaccine to offer protection for more than 10 years, but obviously in a clinical trial, the period of observation would be shorter. We'd like to identify a immune correlate of vaccine-induced protection, which would be great, but we first need to have a trial which shows protection in order to find such a correlate, and then another trial to validate that correlate. In terms of a schedule, if you remember from the COVID-19 pandemic, it makes a big difference whether you've got a vaccine that requires 1 or 2 or 3 doses. So the absolute top end would be a 3-dose regimen, but two would be a bit better than 3, and 1 would be better than 2. Ideally without needing a booster dose at all, but if a booster is needed, then less than 5 to 10 years. And obviously co-administration with other vaccines would be desirable. So I just want to remind you about SARS-CoV-2. I downloaded this figure in March 2023, and at that stage, there were 183 COVID-19 vaccines in clinical development. I filtered on phase 3 efficacy trials, and there were at that stage 50. More than 40 efficacy trials had already been completed, involving more than 400,000 volunteers, which resulted in 12 WHO-approved vaccines against COVID-19. So if we can do this for COVID-19, surely, you might ask, we can do this for TB. But there are some really critical differences. The first one, of course, is that although we went from sequencing a virus in February of 2020 to authorizing a vaccine in December of that year, there were already some things in place. And probably the most important thing that was already in place is that there was a pre-existing lipid nanoparticle mRNA platform for other viruses like influenza. But probably the most important difference is that COVID-19 was perceived as a major imminent global threat. And somehow, although tuberculosis is perceived as major, it is strangely not imminent and somehow less threatening. And the result of that is we saw this avalanche of funding for R&D for a SARS-CoV-2 vaccine, backed by government pre-purchase guarantees, which allowed developers to do trials and manufacture at risk. We don't see this for tuberculosis. And then the final difference, of course, is that tuberculosis is a chronic disease. It doesn't come in pandemic waves, whereas clinical trials of COVID-19 vaccines could be done in these periods of very, very high incidence, where an efficacy signal could be found in a very short time. So we do not, I'll tell you right now, we do not have 50 tuberculosis vaccine candidates in phase 3. What you're looking at now is the entirety of the tuberculosis vaccine pipeline, and I think there's 16 candidates. And that's if we include BCG as a travel vaccine and a couple of duplicates that differ by very minor characteristics, and a few vaccines that have essentially been stuck in this pipeline for a decade. There are some pluses to take away here. And I think one of the pluses is that there's a pleasing diversity of vaccine platforms. So in the pale orange, you'll see that on the left-hand side in phase 1 and phase 2A, there are 3 viral vectored vaccines, and those are mostly being developed for respiratory tract mucosal administration. In the red are mycobacterial vaccines that are inactivated, so killed mycobacterial products. Most of those are not moving forward in the pipeline any further. We've got in the dark orange in phase 3, we have live mycobacterial vaccines, which we'll talk about in a moment, including one live M. tuberculosis called MTBVAC. Then scattered all the way through the pipeline in green, we've got protein adjuvant, traditional adjuvanted protein subunit vaccines, and then the new kids on the block are on the left-hand side, 2 mRNA vaccines, one called BNT164, which comes in 2 flavors, which is now in phase 2A, and then a brand new vaccine, which I know very little about, which I think is from a Russian farmer. But there are a few other things to take away from this pipeline. I mentioned that there are few candidates, but there are also very few novel antigens. So ESAT6, CFP10, antigen 85, those are all very common to many of these vaccines. But probably the defining characteristic of this pipeline is that it's the mirror image of what you would expect a pipeline to look like. So you'd expect 10 times as many vaccines in phase 1 as there are in phase 3. And that's not what we see here. We see quite a few vaccines in phase 3, which is potentially a good thing. But there's very little happening in phase 1 and few new entrants, which is a major problem. We don't see efficacy from the current round of phase 2-3 trials. And that is due to a funding lag, which restricted the entry of new vaccines coming out of preclinical into clinical, which was due to something I'll show you now. It's a consequence of what I call the roller coaster of TB vaccine efficacy trial results. It has its ups, but it has its downs. And when the field has to absorb negative results, sometimes those consequences are long-standing, and that's what you see on the small number of candidates in the pipeline. So more than 10 years ago now, we published the results of a viral vectored vaccine called MV85A, which was the first new candidate to be tested in infants for 50 years. This was given as a booster dose at between 4 to 6 months of age to infants who had received their dose at birth. So it was a booster to infant newborn BCG. And you'll see from the Kaplan-Meier on the left-hand side there that despite a very strict primary endpoint and a very rigorous trial design. The addition of MV85A to the vaccination schedule offered virtually no additional protection to BCG vaccinated infants. And the response to these results in the TB vaccine community was very traumatic. Particularly, it seemed to funders, which in retrospect seems very naive, because who would expect the very first trial of a new vaccine to be the one that shows efficacy? And I think with maturity now, we expect iterative improvements, and you would expect to fail a certain number of times, but to learn from failure and fail well. And then over the subsequent decade, we've had a spate of new results, again, both positive and negative. Here's one of the positive ones. So you might have asked when you looked at the previous slide, well, if you can't boost infant BCG vaccination early in infancy, well, maybe you can give BCG again. And yes, we had this idea that one might give BCG again, BCG revaccination to IGRA negative adolescents. And in this trial, we revaccinated a relatively small group of South African IGRA negative adolescents. I think there were 900 individuals in this trial. And we found that although we didn't affect the rate of IGRA conversion, the rate of sustained IGRA conversion was reduced by almost half. And to explain that, I find it easier to understand by looking at the figure, the two figures on the top, which show that relative to placebo, in which 25% of IGRA conversions reverted, amongst the BCG recipients, almost half of the BCG revaccination recipients reverted their IGRA conversions, meaning they had a lower rate of sustained conversion overall. So unfortunately, IGRA conversion is not bad for you. TB disease is the ultimate endpoint. So what we wanted from this trial was a signal to progress BCG revaccination into a prevention of disease trial. But funders wanted more data, and so we essentially did this trial again with more sites and more participants and with a wider age band, hoping for a stronger efficacy signal. Unfortunately, that was not the case. So here we see absolutely no efficacy whatsoever of BCG revaccination in this larger trial that was intended to validate the signal. We might ask why. So the confidence intervals do overlap to a certain extent, but if there is efficacy, it's in the range of 10 to 15%, which is not feasible. So unfortunately, that trial of BCG revaccination to prevent disease in IGRA negative adolescents, that's probably never going to be done unless someone can reinvent the idea in 20 or 30 years time. Funders like to fund things that work. This was something that I paraphrase, but this essentially is what Jeremy Farrar said at the last Global TB Forum in Brazil. And that is so very true. And I think it's illustrated by this candidate M72 ASO1E, which was the first protein subunit novel tuberculosis vaccine candidate to be tested this time in IGRA positive HIV uninfected adults. And it was tested in IGRA positive adults quite simply because of the fact that to accrue enough endpoints, you have to do a trial in IGRA positive people. Otherwise, the trial needs to be 40,000 people large. But at the time this trial was done, it had not yet been demonstrated that IGRA-positive people could be protected by a vaccine. So here were just over 3,500 IGRA-positive adults without HIV who were vaccinated in Zambia, Kenya, and South Africa. They received 2 doses a month apart of M72, having excluded subclinical TB at baseline by collection of sputum. They are followed for 3 years for microbiologically confirmed TB. And there are a couple of things to take away from that figure in the middle there. The first is that we see very few cases for the first year, and that's almost certainly because the subclinical cases were excluded at baseline. And then the second thing is that we see quite convincing evidence of efficacy in the range of almost 50%. That doesn't seem to wane 3 years out from vaccination, so that's probably quite important. But the other important thing is that this just meets the WHO preferred product characteristics for efficacy for a new vaccine for IGRA-positive adults and adolescents, which is very exciting. And eventually, it took 7 or 8 years, but eventually this result enabled a much larger trial to be funded, and that trial is now fully enrolled and in follow-up of 20,000 adolescents and adults aged between 15 and 44, including a small number of IGRA-positive and a small number of people living with HIV, but essentially the same kind of study population as we saw before, who have been followed up for up to 5 years for microbiologically confirmed TB. So this phase 3 is potentially, if successful, the first licensed TB vaccine that we will see. But it's not the only one. And I mentioned that live attenuated M tuberculosis candidate, MTB-VAC. And this is a particularly interesting candidate because it's live and it's only got 2 gene deletions to virulence factors affecting production of secretory proteins and cell wall, the bacterial cell wall. It contains essentially all the antigens that M tuberculosis has, that M bovis does not, plus it contains all the antigens that M bovis lost when BCG was developed. So it's thought that antigenic diversity and including ESAT6 and CFP10 might be important for protection. And this vaccine MTB-VAC has been shown to be safe and more immunogenic than BCG at a comparable dose in infants, and is currently in a phase 3 trial of more than 7,000 newborns, predominantly in South Africa, who are being followed up for childhood TB, and we would expect to have results by at the earliest 2028, probably 2029. That MTB-VAC has also been found to be safe and more immunogenic than BCG at the comparable dose in adults and adolescents, including those both IGRA-positive and IGRA-negative, but it is now currently in a phase 2B efficacy trial in more than 4,000 IGRA-positive adults aged 14 to 44. Again, results expected in 2029. So we're going to have interesting times in the coming 3-4 years. But of course, as I illustrated with the pipeline, if we don't get an efficacy signal, we still need to fill the pipeline with more and potentially better products. And there's a whole roadmap for how we do that, and how we might make those trials and those candidates more efficient. But this all requires funding. And funding is the one thing that the TB vaccine pipeline has lacked over the years. And despite a very strong investment case in both the lives and the dollars that could be saved by rolling out a new TB vaccine. The funding target for TB for TB in general is still only 12% of what it should be, and the funding for TB vaccines is only 10% of that. So a lot still needs to be done there. But hopefully in 2028, 2029, we will be faced with some implementation decisions. And those decisions will hinge on what data we have and what data we haven't. So even if we show efficacy in one of these ongoing trials of M72 and MTB-VAC, we will have, and this is, I'm talking about the adults here, we will have efficacy in IGRA-positive people between the ages of 15 and 44. Well, in safety groups, safety and immunogenicity groups, we'll have data for IGRA-negative people and people living with HIV who are receiving antiretroviral therapy in that same age band. But we will have no data whatsoever on children younger than 15 years of age, on adults older than 45, the elderly, in inverted commas, for those of us who are deemed elderly, on people living with HIV who are not yet established on antiretroviral therapy, on pregnant or lactating women, people with previous TB, and then obviously on specific risk groups who don't necessarily fall within these other criteria. So the question is, how might implementation happen in terms of will the label be restricted? Would rollout be targeted, and to what extent can these data be collected in phase 4? So this is my last slide. So yeah, I've talked a bit longer than I intended, but this is essentially, I think we're entering, this is not the end of the story, but we're entering this final chapter of TB vaccine development. And I hope I've shown you that there's a great need for a new vaccine, particularly for adults and adolescents, that although the pipeline is small and there are very few early phase candidates, we are currently at a stage where the stakeholders are matured and we can resist a negative trial result or two. We do have 2 very promising candidates in trials, and we are expecting efficacy data in 2028, 2029, but there's a lot we don't know, and that will have to be done, or that will have to be studied in post-licensure studies, and that includes can we find an immune correlate of protection so that we don't have to do these massive trials. So I think that's, yep, that's the end of my slides. So I'm happy to take questions and go back if necessary.